Good afternoon, good evening to all of you wherever you are. I'm really excited. This is like a new group for me, so this is really cool to be with some of you for the first time, and um, I'm really excited to be here. So thanks for the invitation. So yes, I'll be giving. I was actually supposed to give this talk in the spring, and I like just basically became deathly ill, and so this is the reschedule of it. So I'm really excited to be here, and I'm excited to be healthy and being here. I'll be giving this talk on what makes a cyanobacterial bloom disappear, which is a paper that I co-led with Ted Harris at Kansas State University Biological Station. So I'm just gonna jump right into it, and I hope you enjoy it. Okay. Um, let's see if my things will work. Okay. I think it's gonna work now. So I always just kind of include this. So when I say that I work at the Lake Superior National Estuarine Research Reserve, people often, especially if you work in freshwater systems, say, "What is that?" Or maybe if you're not a U.S. based person, you're thinking, "What is that?" So the National Estuarine Research Reserve System, or what we call the NERS, or sometimes the NEARS on the East Coast, is a network of 30 coastal sites that are designated to protect and study estuarine ecosystems. And it was established through the Coastal Zone Management Act. So the reserves represent a partnership between NOAA and coastal states, where NOAA provides the lion's share of the funding and national guidance, or essentially our marching orders. And then each site is managed on a daily basis by a state agency or university. So in the case of the Lake Superior Reserve, which is located here, it's University of Wisconsin-Madison Division of Extension, which I don't fault you at all, Sarah. It's quite the mouthful. And so we get a lot of input from local partners, um, and uh, and also from NOAA as well. So the research reserves cover about 1.4 million acres of estuaries and are focused on stewardship, research, training, and education through our program. So it's kind of a unique blend of all of these things happening all under one roof. So we're not just a research institution, but we're also doing a lot of these other things. So like I said, I'm going to be giving a talk on this work that was published, I think in March, in Harmful Algae. So before I dig into what this paper was about, I want to acknowledge all my co-authors and highlight the really collaborative nature of this work. So as I said, I co-led this work with Ted Harris. You can see his name up here at Kansas State. But this work included 28 authors that were from 3 different continents, 11 different countries, and 24 different institutions that include both academic research and management agencies. And all of this work occurred through the Global Lake Ecological Observatory Network, also known as GLEON, if you're familiar, as part of the Cyanobacteria Working Group. And so I know you're all really excited to hear about phytoplankton. That's what we're all really pumped about. But if you'll just humor me for a moment, I just want to talk a little bit about GLEON and this kind of work that GLEON does, because I think in a time where it can feel like it can be hard to collaborate across various boundaries, whether they're geographical or political or institutional. It's really great to highlight these success stories of people coming together from a really diverse group and accomplishing some cool science. So I think it's just really important to highlight that component of the work and not just kind of leave it as a footnote. Um, the mission of GLEON, or the Global Lake Ecological Observatory Network, as I said, is to conduct innovative science by sharing and interpreting high-res sensor data to understand, predict, and communicate the role of lakes and global climate change. And this was kind of the original mission, but over the years, it's really expanded to include other types of research, including like large-scale collaborative experimental work, reviews, and important topics, knowledge gaps, and more. And GLEON includes a network of more than 500 members from 49 different countries and counting. Um, so over the years, as I said, the mission has really kind of like morphed and changed. And so now this is kind of the work in progress, but it's still networked lake science. But what that means is a little bit different. Um, and the Global Lake Ecological Observatory Network is conducting international team science to advance our understanding of lakes and their benefits in a changing environment. So originally, it really started out as like this network of high frequency sensors, but as you can see, it's really kind of changed to be a little bit more holistic, essentially. So here's just a little bit of a sampling of some of the work that GLEON does. So sometimes they have papers that are just on describing the network, which is it's really unique. It's like it's a grassroots network. It's very bottom up, and one of the really unique things is the bread and butter of Gleon is that it has these different working groups. So when you go to a Gleon meeting, you're not listening to talks, you're going to different kind of topical work groups. So I really like cyanobacteria. So, I hear about the different work groups and what they've got going on, and I can bounce around for the week. But essentially, you're going for the week, going to the week, and you're doing what you love most and like talking science with people who are like into things that you're also interested in. I think one of the kind of funniest analogies that I've ever seen for that was the equivalent of like scientists saying, let's start a band is let's write a paper together. So there's a lot of bands getting started at Gleon, which is really cool. And sharing of information and collaboration, which is really unique. There's topical reviews. So like the one that I'll be sharing today, we've done a few of these out of the cyanobacteria working group. And this might be another one that's of interest to this group is like such as the role of organic nutrients and phytoplankton communities and many others. Um, there's also coordinated lake sampling. So these papers that often you see like really enormous author lists are, these opportunities that Gleon puts out to come together and like, try to do things at like a really large geographic scale. Um, and also the social science aspect is being brought into some of the work that's being done, which is definitely a big development or a big change from the original mission of Gleon. Teaching is also a big part of Gleon. There's a lot of folks that are educators in some way that are involved in Gleon, and I'm sure many of you too. And that's also definitely brought into the Gleon model. And actually, a pretty significant portion of the Gleon membership is students and early career folks. Um, and then lastly, Gleon also does a lot with modeling efforts and partnering modeling with like your experimental and field data collection folks. So I just wanted to put that out there because this work, it really is a Gleon effort, and I don't want to, I guess, like undercut the importance of that. And like I said, the collaborative nature of the work that has been done and the work that I'm presenting today and other projects that I've been involved in Gleon. So I think the website is a little bit under construction right now, but I think that the one at the top should work. Otherwise, you can always get into contact with me if you want to learn a little bit more about Gleon. Okay, so thanks for humoring me while I kind of went on that little side path. We'll get back on track here and talk a little bit more about this loss processes work. So this was born out of the 2022 Gleon meeting that occurred in Lake George with the Cyanobacteria Working Group that had 29 people from 10 different countries at that time. So during work group discussions, it was noted there's just a lot of focus on what causes a bloom, which there's good reason for that. So often we're thinking about like, okay, maybe if we can prevent what causes a bloom, then we can nip it in the bud, essentially. But there's not a lot of focus on loss processes, and that just kind of either gets ignored or just certainly not given nearly as much attention because we're just, like I said, often thinking about the front side of this equation and like what's causing growth and like how do we prevent that. And so that really led to this paper that I'll be talking about today. And another component of that is you may or may not have seen this other paper that I led along with Ted and others from the Cyanobacteria Working Group called Blooms Also Like It Cold, where we documented cyanobacterial blooms occurring in cold water conditions. And you've all probably been through this process of getting a review back on a paper and you know trying to dig through how to incorporate those things, et cetera. And one of the comments that we actually got back on the review for that paper was, what about the effect of loss processes and the effect of temperature on loss processes? And that also contributed to this idea of like, what about these loss processes? Let's think about this. What do we know about them? Can we like really kind of synthesize what we know and put it together in a nice review? So those kind of two things are what really motivated this paper. So I'm sure all of you know about algal blooms, but just in case there's like a student on here or somebody who is just like, you know what, I'm going to do something crazy today and I'm going to go learn about algae. I'm going to go over this really quickly because there's usually at least one person who's like a little bit shaky on the information. So broadly speaking, an algal bloom is an accumulation of algal biomass, and it's often observed as a green scum on the surface of a water body. And they're often the fallout from excessive nutrient inputs, allowing unchecked growth of algae or phytoplankton. And this can be problematic problematic for a few reasons. Blooms can block sunlight needed for other organisms, and as they die and decompose, oxygen is used up in the decomposition process, causing other organisms to be stressed or die due to a lack of oxygen. So, a really classic kind of example of what can happen is like a fish kill. So, there's really kind of like famous photos of many lakes, including Lake Erie, for example, where algal blooms dying and decomposing cause bottom water hypoxia or anoxia, resulting in fish kills. So that's kind of the overarching, like, what is an algal bloom? Why is it problematic? And then we have cyanobacterial blooms. So cyanobacterial blooms cause all the same ecological problems that I described in the previous slide, but then they have this added issue that they have the ability to provide or to produce toxins, namely liver, neuro, and dermal toxins. So neurotoxins, hepotoxins, and dermatoxins. And when these are ingested either through breathing or drinking, they can cause severe illness and death. The PSAs that are put out are usually often focused on like children and pets because they're more likely to kind of go in the water and indiscriminately drink it or accidentally drink it. But it still can make pretty much anyone sick. So you might have seen articles in the news where you are, or maybe in other places where a pet died after drinking water that had cyanotoxins present, or, you know, cattle deaths drinking from a pond where there were cyanotoxins present, and even in some cases, children or adults that got severely ill from ingesting these. So cyanobacterial blooms are kind of problematic in the same reasons as before, but also they pose this pretty significant public health risk. And so we often call cyanobacteria blue-green algae due to their pigments that they contain. And sometimes when you see a cyanobacterial bloom in the water, it actually does look very blue-green, but often it also just looks green, similar to your eukaryotic or phytoplankton algal blooms. Um, and so we also call them algae, but really they're not algae. They're cyanobacteria, as the name suggests, are actually bacteria, but they also do photosynthesis. So their mechanism for doing photosynthesis is different than their eukaryotic counterparts. So for example, instead of chloroplasts, cyanobacteria have phycobilisomes and accessory pigments that allow for more efficient light harvesting than traditional phytoplankton. But essentially, they're still harnessing the power of the sun for growth and reproduction. So to the naked eye and like to, for general purposes, they're kind of operate in the same way, but once you get down to the cellular level, they're actually very different. Um, and I have to just put out there that cyanobacteria get a really bad rap, but they're also a healthy part of all ecosystems. So they've been around for billions of years. You can actually thank them for the majority of the oxygen that we have in our atmosphere. Um, and so there is kind of, I think, this misunderstanding, or they're a little bit of a misunderstood plankton, because just because they're there doesn't mean anything bad per se. But when we get this abundance of growth, and especially when we get toxin production in places where people are interacting with the water, that's when we start to have problems. So they're not an inherently bad thing. There's just situations where like unchecked growth and production of toxin can be problematic. So now that we've laid some of this groundwork, we can get into this growth versus loss. So when we started this paper, we thought it was pretty straightforward to talk about growth and loss, but really quickly we got kind of like tied up in limitation, and how do you disentangle limitation from loss? So we had to really kind of go back to our basics and define the difference between these processes. So we have growth limitation. So this is related to factors that affect cyanobacteria population growth rate, and it happens when there's a deficit in essential resources such as light, nutrients, and temperature, or bottom-up regulation. Loss processes, on the other hand, are anything that leads to a reduction of the standing stock of cyanobacterial biomass. So blooms form when the net population development is positive, or basically when your population growth rate exceeds the rate of loss processes occurring at that time. And then when the reverse is true and losses are overriding growth, blooms can stop developing and accumulation may slow down. Um, so I went through this really quickly, and I'm going to have another slide that maybe for you math-oriented folks is a little more helpful in thinking about this, because even when we were writing this up, like it was still felt hard just to, like, I guess, verbally or just in writing, really disentangle these things in a really clear way. But when you go to the modeling literature, it's actually broken down in a really nice way. So let's take a look at that. Okay, so Reynolds in 1984 compartmentalized loss processes into hydraulic washout, sedimentation, grazing, and death, and this was represented by the following equation. So you have your specific growth rate, K prime here, and then you have exponential growth, and then your loss processes. So you can see in these equations, growth and loss are specifically broken out into two different things. Similarly, Chopra 2008 presents biomass concentration as a function of time as growth over time, your dA over dt. You have K sub G, which is your growth rate as a function of temperature, nutrients, and light, which are your limitation factors. KD is your loss rate, so D meaning death essentially, and then your A is your algal biomass. And Kg specifically is your temperature. You have your temperature specific growth rate and then nutrient and light limitation factors. So these really, I think, help demonstrate how growth limitation and loss processes are really separated out from one another. So limitation is like basically kind of like a check on your resources, your temperature, your nutrients, and light, whereas your loss processes are specifically like removal or taking away through some process. And that, like I said, it's really kind of like nicely broken out. In the modeling literature and in the equations. Okay, so this is our graphical abstract from the paper, and will be kind of a roadmap for the talk. And so we have our growth shown here, and I hopefully you can see my mouse, but if not, you have your growth over here, kind of outlined in green on the left, and then we have our loss processes outlined in orange here to the right. And we're going to focus on the loss piece of this because, as I said, there's been a lot of focus on growth, and maybe we can cover that another time. Okay, so we'll first start with physical transport and dispersion. So that's going to be down here. So we have our flushing and our mixing. Um, so when we have flushing, it can cause hydraulic washout, which removes cells from the system. So hydrodynamics in general play an important part in whether a bloom disperses or is formed. So you may hear that kind of classic example of like, oh, we had a really calm day, and then cyanobacteria, which have the ability to be buoyant, rose up to the surface, and then light breezes blow them in, which you can see in this graphic here, and then you have a bloom. So sometimes a bloom is formed just from this like essentially water movement. Well, the same thing can happen too in the opposite direction. So there's those classic examples of a bloom was there this morning and it was gone by the afternoon, which is something that we often see in Lake Superior, is they can be really ephemeral depending on what the wind conditions are. So this could be a small lake or an embayment. It's just, kind of like your area of interest or your discrete area where you can have an accumulation and you can have it come out. Washout can also be caused from like a large storm event or rain event. So a lot of wind or a lot of water moving from the system can really like flush particles, including phytoplankton and cyanobacteria, out from a system, which can cause the loss of a bloom. And you can see in this graph here on the bottom, when flushing is high, cyanobacteria low, and vice versa. So mixing is a little bit different. So we're kind of thinking more in the horizontal scheme in the previous slide, but here we're thinking more in the vertical. So two things can happen here. One, mixing can push cells down to depths where they just can't overcome density gradients, or in the case of, and then they sediment out of the water column. Um, and then a second thing that can happen in the case of cyanobacteria that have gas vesicles that allow them to be buoyant and access different resources at different depths in the water column. If they're pushed down far enough, that increase in pressure with depth can overcome their gas vesicles and cause them to collapse from turgor pressure, and then also cause the cells to sediment out of the water column. So sometimes mixing with cyanobacteria in particular, it can like push them down and disperse them, but then they can become buoyant and come back together. But if they're mixed down far enough, those gas vesicles can collapse and then they no longer have the ability to be buoyant, and then they will sediment out and die as a loss process. Okay, so now I'm going to move on here to oxidative stress and UV light. So the primary energy source for photoautotrophic organisms such as cyanobacteria and other phytoplankton is photosynthetically active radiation, or what we call PAR. And photoautotrophic organisms produce reactive oxygen species, which are shown here in this kind of like red, I don't know, explosion graphic. And so it's a byproduct of reactive oxygen species, or ROS, are byproducts of photosynthesis, but they can cause oxidative stress when there's overproduction. So oxidative stress can be toxic for cells and lead to cell death. And so there's some examples here. So in the case of lipids, you can see chain breakage, induction of membrane fluidity, and permeability. For proteins, it can result in modification of amino acids, fragmentation of peptide chains, inactivation of enzymes, and in DNA, we can see strand breakage, removal of nucleotides, modification of bases, DNA protein crosslinks. So basically, it can cause a lot of damage to the physiology and the cell structure, which can ultimately lead to cell death. Um, Light also plays an important role in the formation of reactive oxygen species, with high amounts of UV light promoting overproduction of reactive oxidation species, or reactive oxygen species, excuse me. Um, Something that's really kind of interesting about this, though, is that you can have species level differences, but you can also have strain level differences in your responses to light. So, cyanobacteria, in comparison to eukaryotic algae, have been shown to have significantly higher sensitivity to hydrogen peroxide, which is a reactive oxygen species. But then also there's the strain level difference within cyanobacteria. So you can see in this figure over here, the wild type, which is containing microcystin, which is a really common cyanotoxin produced by microcystis as well as others. And then you have this mutant, which is microcystin deficient. So these are two different strains, one containing cyanotoxins essentially, and one not containing cyanotoxins. And the species of microcystis that is not containing cyanotoxins is actually showing a much higher sensitivity both to light intensity and also exposure to hydrogen peroxide. So there is that kind of species level difference in response to reactive oxygen species. And then there's also that strain level difference, and that the presence of cyanotoxins in this example actually has a pretty uh, important role in the level of like resilience essentially of cyanobacteria to reactive oxygen species. Um, Similarly, solar UVB radiation, so there's UVA, UVB, and UVC. UVB radiation has been shown to have negative effects on cyanobacteria, leading to effects on growth, pigments, enzymatic processes, buoyancy, DNA, and protein structure. So very similar to those reactive oxygen species effects. So um, this can be a really important loss process that ultimately is leading to cell death and removal of the standing stock from the population or the bloom. Another biological, biochemical, if you will, loss processes is these allelochemicals. So these can be produced by many different sources. So there's macrophytes, there's fungal, there's bacteria, there's cyanobacteria, and then there's even other eukaryotic phytoplankton that can produce allelochemicals that impact cyanobacteria. But the example that I have on here is specifically for macrophyte, and that's probably where most of the literature or information is right now in the literature. So allelochemicals are chemical compounds that are produced by many terrestrial and aquatic organisms, and they can have both positive and negative effects on other organisms. Of course, since we're talking about loss processes here, we're thinking a little more on the negative impact side. Um, but allelochemicals that affect cyanobacteria are produced, like I said, by macrophytes, heterotrophic bacteria, fungi, phytoplankton, and other terrestrial plants. And what these chemicals do is they can disrupt photosynthetic pathways, but they can also disrupt physiology and growth. So kind of similar in a lot of ways to reactive oxygen species. And also similar to the effects of reactive oxygen species, you don't see the same level of sensitivity across species. So for example, Planktothrix has been shown to have more resistance to allelochemicals from macrophytes, while Oscillatoria, Dilocospermum, and Microcystis can be highly sensitive. So it's really not a one size fits all, but you have a really nice graphic here. So you can see these macrophytes, which are these plants on the bottom here, and then the cyanobacteria, which are basically these straight green rods. Um, the allelopathic effects are these red arrows. So in the macrophyte dominated, or I guess macrophyte present scenario, you have these allelochemicals that are coming off of the macrophytes, which are essentially controlling algal growth and kind of keeping a more balanced ecosystem. Whereas in this phytoplankton dominated system where macrophytes are not present, you don't have that kind of check and balance on growth. And so you get, a lot more growth of phytoplankton for two reasons. One, you don't have the allelochemicals that are basically tamping down growth. And then you also have a loss of competition for nutrients, which is really more on the growth limitation side of things. Um, so we also have infections. So Sarah talked at the beginning a little bit about chytrids, so that falls squarely under this group. So we have kind of three main categories that we're thinking about here. We have bacterial infections, fungal infections, and viral infections. So mechanisms of bacterial infections and effects can be really variable. There's often a lot of like coevolution of bacteria. So and this is also similar with the allelopathic, where bacteria can be helpful and they can also be harmful to cyanobacteria and other phytoplankton. So in some cases, they can cause cell lysis, they can cause reactive oxygen species production, they can disrupt physiology, they can cause programmed cell death, which I'll talk about in a minute. So they really have kind of a lot of tools in their tool belt to disrupt phytoplankton growth. And like I said, a lot of this is probably really dependent on the microenvironment around a bloom and whether those bacteria co-evolved with the cyanobacteria in that community or other phytoplankton in that community. Um, for fungal infections, you have zoospores that swim out from mature sporangia that are developed on infected phytoplankton hosts. So not just cyanobacteria, this can be other phytoplankton as well. And then they use chemotaxis to find and attach and infect new hosts. So one, there definitely can be differences among species, but there also can be a lot of environmental factors that affect actually all of these infections. So for example, cooler temperatures are usually going to lead to lower infection rates. And in some cases, even if it's not necessarily lower infection rates, it can reduce the number of infections that lead to ultimately cell lysis or death of phytoplankton and cyanobacteria. Um, and then viral infections, they often follow the seasonal patterns of the host. So when you there's not really kind of like anything it's particularly special about viral infections, just that, as I said, they kind of tend to follow the seasonal succession. Or like maybe slightly lagged from them. So these are all something that we see as a loss process both for cyanobacterial blooms, but also for phytoplankton blooms in general. Next up, we have grazing. So there's lots of things that fall under this category, and probably one of the things that most often comes to mind when we think about loss processes, or at least it's one of the first things that came to our group's mind, probably behind physical mixing and flushing. So there's many different types of grazers that can impact phytoplankton blooms and cyanobacterial blooms. So there's zooplanktons, mussels, mixotrophic algae, ciliates, and others. Um, Consumption of bloom-forming cyanobacteria is a primary loss that controls cyanobacteria or bloom phytoplankton dynamics, but cyanobacteria traits such as toxins, polyunsaturated fatty acids required for herbivore growth, and morphological defenses like colonies or filaments can often reduce grazing loss, and that's something that's more specific to cyanobacteria. Um, Cladocerans especially are large-bodied organisms such as Daphnia, and they have the highest potential to graze down cyanobacterial blooms, mainly due to their size and the combination of traits that the genera possesses. But studies have also found that the interaction between cyanobacteria and zooplankton is just really complex, and factors such as genotypes, coevolution of the grazer and the cyanobacteria population can really impact the potential. And overall, copepods are just not a strong candidate for cyanobacterial loss via grazing. However, again, that coevolution aspect or locally adapted populations might be able to graze on filamentous cyanobacteria more readily when nutritious prey are not absent. So you may have seen in the literature that cyanobacteria are kind of like not the most desirable food essentially for zooplankton, and that's I think seen a lot. But when there's not a whole lot of food abundant, then and it's really kind of the only option that's there, you'll see more grazing. And again, temperature can affect these interactions as well. So when you have warmer temperatures, you tend to shift to more generalist grazers of zooplankton, whereas in cooler temperatures, you tend to shift towards more selective feeders. So essentially, you can think of that as under warmer conditions, you have like not picky eaters, and under cooler conditions, you have picky eaters. So when things are warmer, it's probably more likely that cyanobacteria would be grazed on. And in cooler temperatures, it's more likely that more nutritious eukaryotic algae would be grazed on if they are available. Um, so in addition to or in addition to zooplankton, there's also filter feeders such as mussels and bryozoans. So mussels have been coming up a lot with the invasion of drysenids that are non-native. So they can have a really strong influence on planktonic species distribution through grazing. But again, this is really species specific, and some studies have even found increases in cyanobacteria after the introduction of mussels because they're preferentially grazing on more nutritious algae, essentially. And then lastly, fish can also be a significant role in grazing cyanobacteria, both directly and indirectly. So they can directly graze, and there's some really nice biomanipulation experiments from Lake Taihu using silverhead and bighead carp, where they were added to mesocosms, and they did actively graze on cyanobacteria. But then there's also the trophic cascade. So the way that food web structure interacts to either promote or tamp down zooplankton can ultimately impact the phytoplankton population. And specifically, in this case, we're thinking about cyanobacteria. So there's a lot of different kind of organisms that are out there that play into grazing, but I think it's very complicated by the effect of one, what is the food web and the trophic cascade look like in that system, and also what are the environmental conditions around that are, I guess, going to kind of like shape those relationships between the various components of the food web. And then the last one that we have in our loss processes here is akinates and programmed cell death. So resting stage cells and programmed cell death, also called PCD for short, are defined losses because they remove cell from the water column. So they're dying, they're sedimenting out, and then they're removed from the water column. So it's again that loss of standing stock. Resting cells are have been shown to be spurred by decreases in growth limiting factors. So they are kind of at least related to the limitation piece, but ultimately when an akinete is formed or an akinete, both ways of pronouncing are acceptable. Um, when they're formed, that's still just like a direct loss of vegetative cells from the population that's not going to be reproducing. So it's still considered a loss because then an akinete will sediment out to the bottom of the water column and hang out until conditions are better for germination. Um, Programmed cell death, also known as regulated cell death, refers to non-accidental cell death in response to different internal or environmental triggers, and growth limitation can help accelerate resting cell production and programmed cell death loss processes. So they're not completely exclusive from one another. So PCD and cyanobacteria has mostly been studied in cultures, and it's often triggered by really atypical conditions. So really high salinity or the presence of oxidants or herbicides. So, like really kind of like particularly odd conditions. But as I said, most of this has been studied in a culturing environment, and there's not a whole lot of information about how this works in a normal natural environmental setting. And then I have a bonus for you. So there's also the interventions that we do to control blooms. So there's kind of like the way I broke things down before is like physical, chemical, and biological. Similarly, we have kind of like mimicked or harnessed those processes for bloom management strategies. So um, physical methods for creating loss for a cyanobacterial bloom or a phytoplankton bloom include aeration, mixing, and then inflow and outflow manipulation. So in some cases, if you have control of a water system, you can like flush out the system. But in general, the size and morphology of the system, as well as the infrastructure that's actually required to do those things, are really going to like determine whether it's appropriate or effective. So I work on cyanobacterial blooms in Lake Superior. We're certainly not going to install aeration or try to flush these systems out. Whereas like maybe in a small pond or a small lake, some of these methods might be a little more effective. Chemical approaches include the addition of flocculants and coagulants, which are also used for phosphorus reduction, as well as the use of algicides when a rapid reduction is required. So flocculants and coagulants are designed to remove cyanobacteria through the addition of material that adheres to the cyanobacteria, similar if you're familiar with like phosphorus removal, and causes them to sink out of the water column essentially. So that's that sedimentation piece. Algicides, on the other hand, are most common are really kind of used for like that. We need to do something really quickly and just like kill them off. So some of the most common algicides for reducing cyanobacteria are copper-based. So copper sulfate has been used for controlling blooms for over a century. And then lastly, there's biological treatments, which is essentially kind of food web manipulation, and that's not a new concept at all. But there are cases where biological manipulation is being used to try to control the trophic cascade to reduce cyanobacterial blooms, and I would imagine this has also been used for other types of phytoplankton blooms as well. Again, this really kind of depends on the system that you're working in. So, what is the existing kind of food web structure, and also how large is the system? So, again, using Lake Superior as an example, we're not going to just kind of like, introduce fish that are and expect that it's going to be effective at the size of a lake like Lake Superior. So while we're often really focused on this kind of like growth piece, the loss piece is really important, and then we're also thinking about like how do we think about this going into the future? So I'm not going to say a whole lot on this because, of course, we're thinking about climate change a lot, but we kind of came to the point when we were working on this is like, at what point are we kind of speculating wildly, which can be fun, don't get me wrong. And I often like to do in talks because it can be kind of a fun venue to do that in. But it's really hard to say because some of the things that are going to increase loss processes or going to decrease or increase loss processes are also going to decrease or increase growth. So take temperature as an example. So you might see an increase in temperature, which is going to increase growth of cyanobacteria or phytoplankton in general. And let's take grazing as an example. When you increase temperature, grazing rates tend to increase, and also you tend to shift towards those more generalist grazers, as I said before. And so um, it's really hard to try to like understand or try to, as I said, speculate about how these things will interact with one another uh in a future climate or a warmer climate. Another example is like, okay, maybe let's think about like increased storm events or rain events. So you might see increased flushing, but that might be really a temporary effect on blooms because you're also introducing more nutrients into the system when you have these storm events, which could be a positive on cyanobacteria. So if you want to check out the paper a little bit about the things that we talk about and the challenges in trying to think about that, I would definitely encourage you to do that. But I just wanted to note like how complex these interactions are and how difficult it is to try to pre predict or project how loss processes are going to change and how that might impact blooms into the future. And, with the complicating factor of how those same processes are going to impact growth, and that loss and growth often move in the same direction with temperature, for example. Okay, so I'm going to wrap up with some takeaways, as is kind of customary for these things. So cyanobacterial bloom dynamics are governed by a balance between growth and loss processes, and so I would encourage myself and others to really to be thinking about both sides of the equation, especially when we're thinking just from like an ecological context, and not kind of like just focus on growth and kind of forget about the loss, but to really think about both pieces of that. Considerable attention has been given to these processes that promote cyanobacteria, and we should spend a little bit more time essentially studying loss processes or thinking about them or including them in our studies. So thinking about the entire life cycle of a bloom. Um, Cyanobacterial traits and in situ physiochemical or environmental conditions can affect how and when these loss processes can override bloom processes. So that balance between growth and loss to make a bloom disappear or reappear. And then just in general, a deeper understanding of loss processes affecting bloom will give us a more holistic understanding of bloom ecology, which can improve modeling, prediction, etc. So with that, I'd like to thank you for your time. I'm really grateful that I got to be here and talk with you all. I wanted to put the link in the chat, but I also don't want to pull this down. So I'll put it in the chat when I close this out, or if anybody's able to throw it in there for me, that would be really great. Again, I want to acknowledge my co-lead on this, Ted Harris and Gleon, for being behind this project, and I'll take any questions that you have.
Great. Thank you so much, Kate, for a really fascinating talk. If anyone has questions, you may go ahead and raise your hand or put it in the chat, and Marina can read it from there. So either way, either hands or in the chat is good. And maybe while you're thinking about your question, I want to Um, really appreciate your talk, Kate, and um, you know, you touched on this a little bit. Sort of the obvious question is how much are increased incidence of blooms actually due to increases in loss processes? Um, if you want to speculate wildly, for example, higher temperatures facilitating fungal and viral predation, um, sort of tipping the scale as you showed it in the other way.
Yeah. So like I said, it's really hard. I think it's hard because I've so when we talk about blooms, there's a kind of this like inherent management piece to it always, and like we're always wanting to kind of speak to that management perspective, and that always kind of calls for this boiled down view of ecosystems. And I know people get so frustrated with scientists because we can't just give you a straight answer and stuff, but I kind of push back on that a little bit because it's often just not a straightforward answer, and it's very hard to generalize over large, you know, varieties or ranges of systems essentially. So I think it really is pretty system dependent. So, in a system where you might have a lot of chytrids present, you're going to get a very different outcome um, by changes in temperature, right? So you might have like higher infection rates. So you might have higher infection rates, but it also depends on your species that's there. So cyanobacteria that we see in Lake Superior in particular is Dilocospermum lemmermanii. And this species has a really wide temperature optimum and like a wide like kind of like temperature plasticity. So the range of optimum temperatures can be as low as 13 degrees Celsius up to 20 degrees Celsius, where oftentimes people are thinking about cyanobacteria, they're thinking 25, 30 degrees Celsius as optimal temperatures, but you actually start to see a drop off or a shift to other cyanobacterial species. So you might like kind of bringing that all together. So you might see an increase in temperature, but you might have a species such as in Lake Superior of cyanobacteria where it gets warmer than what's optimum for that species, but maybe is still like promoting infections. And so those kind of like really cellular level things are really important for understanding this like kind of like ecosystem scale. Um, and I think it's really hard to, yeah, kind of give like a one size fits all because, for all of us that are limnologists, each lake is like our own special, like, entity in and of itself. But I think that's also kind of just true, and it's really hard to give that kind of like general answer. So that's my typical
Yeah,
science non-answer.
no, that's good. And I think Lawrence in the chat has asked, at least the way I interpret it, sounds like a similar question. Um, are there essential benefits of having loss in cyanobacterial processes? For example, lakes or aquatic systems in relation to global changes. I'm not sure if I understand that. Lawrence, if you want to clarify that question at all, um,
We'll see.
go
He's
ahead and drop that in. Maybe just to follow up then, given that these systems are so complex, what do you see as is it is it just say productive? Is it productive for us to work at predicting um, blooms if we are not characterizing um, all the parameters, except as you say, maybe in um, a local system with gliotrichia or understanding
ooh, yeah. Oh,
in a much more narrow context.
you're catching me in a trap, Sarah. So it's funny because I talk a lot with modelers. So like I have a little bit of a history in modeling and I did some modeling as part of my PhD. And so I have a couple of thoughts on that. Um, I think we've really gone too far in the direction of like building like really crazy specific models. I think, of course, like kind of backing up, kind of the answer is like depends on what you want to do with your model, right? Like that's always kind of one of the questions in your model development. But I think models, there's two things. Like you develop these models that have, 30 or more like state variables and 30 or more like specific parameters that are probably changing over time and are based on like one or two studies. It's really, I think, I don't know that those are super useful efforts. I think when we go to models, I think a little bit more like general models can be helpful and kind of that general understanding basis. In some cases, though, you're developing models to like work with a water manager to make sure that the water is safe to drink. And that's a little bit of a different situation where you want something that's really specific to a system. I think models can be really helpful for like these kind of like broader understandings. And like, I think it's very hard to find a balance when you're developing model between that kind of like broad brush understanding where you're capturing kind of like the main things that are like affecting the system. Um, but I also think that models that kind of find that balance between like being a little bit general, but also incorporating some of this trait information are like very interesting. And I don't know if it's like the, modeling future or whatever. I know some folks are doing that, but I think that that could be really useful in trying to understand that because this trait level information I think has a lot of impact on what we see for outcomes. Um, so I think modeling is really tricky when we're thinking about blooms, and it does kind of depend on what exactly it is you're trying to do with them. Um, like I said, you have the example of like I'm working with a water manager and I need to make sure that the water is going to be safe for the community to drink. And so I need to know whether toxins are going to be produced or when do we expect to see peak biomass, et cetera, versus kind of like, are you looking for a broad understanding over a wide geographic scale? Or, I don't know. I think that's like completely not answering your question, but these are things that I think about a lot when we're developing models, like even for Lake Superior right now is what right now we're at the beginning of thinking about some of this and trying to suss out like what direction do you take this in? Do you drill in really, really detailed species level information, or do you start with this kind of like large area, really general kind of approach? And I think both are probably valid depending on the questions you're asking.
Yeah, I want to invite other folks to jump in by raising got a hand. Go ahead, and you can can't see who it is. Please go ahead and
Eldrin.
ask Eldrin. You can go ahead with
Okay,
your question.
thank you. I'm just wondering about about the microbial sorry, shut up. Um yeah, the part where you said about infections, bacterial and viral infections. I'm
Hmm.
just thinking, at what stage of the bloom does it happen? Because usually, does it happen at the middle or usually at the end stage?
Ooh, yeah, that's a really good question.
And
I don't
then
Go
and
ahead.
then you talk about mitigation strategies. Are you considering using bacteria or microbial as biological control agent for these blooms? Because you talk about
Yeah, okay,
um
so
yes,
I'll answer the two parts to your question. So
yes, yes.
one, I have a friend who's a microbiologist on this paper that can answer your question a lot better. Hans Peter Grossart, he's like my go-to person for this. I don't have a confident answer for you on that. I think oftentimes it's when growth is increasing and you're approaching stationary phase, but I don't know for sure because that's kind of like outside of my direct like kind of wheelhouse for understanding this. Um, but I know, like I said, for viral in particular, it tends to follow the seasonal pattern. So I think that is pretty tightly coupled to biomass. And then the other piece you were asking about, oh, is like the using infections for bloom strategy control. I have don't know anybody personally who is actually doing that, but I have heard of it like pitched before as like, okay, let's see if we can cause cells to lyse using viruses and stuff like that. I don't know if it's like a widespread practice. I don't think it is at this point, but it's definitely something that's been brought up as in theory and probably has been like, pilot tested in places.
Mhm. Follow-up question. Just last one. The program cell death mechanism, does it happen like anytime during the whole stage of the
For
Do you
programmed
know?
cell death?
Yeah, yeah.
Yeah, I
Is
think
it
it can happen anytime. It's, if you have a
uh,
cell, whether there's a bloom there or not, if you have a cell there and you just kind of like blast it with crazy conditions, essentially, it's gonna die. It doesn't matter whether that there's a lot of biomass or there's a little biomass. It's really kind of, that's like at the cell level. It's just like, is something going on in the environment that's catastrophic and it's like just causes the cell to shut down and die.
okay, okay. So it's based on environmental factors.
Yeah,
Okay, okay.
yeah.
Thank you very much.
Yeah, thanks for your questions.
It looks like we have a question from Leah. She said, "Thanks, Kate, great talk. I wanted to ask you why we have in some ecosystems a seasonal peak of diatom growth before the cyanobacteria peak."
Yeah, so this is kind of like the the peg model, right? Essentially. So, in the spring you have silica available, and so diatoms are, and they're also, and you also have cooler temperatures, which are more associated with diatoms. So diatoms in the spring have an abundance of their resources that are available, so they need silica in addition to other nutrients. So they're going to take up the silica and they're going to grow until they deplete the silica stores in a system, and then they're going to be replaced by chlorophytes or your green algae. And then they move on to cyanobacteria later in the season, when temperatures are warmer and there's changeover in resources. So you have kind of like, oh man, maybe this is like really testing all my like limnology textbook knowledge right now. So you have your spring diatom peak, you have silica abundance, your diatoms grow, they use up the silica, then your chlorophytes come in and grow. Then your zooplankton, because they have a higher standing stock of food available, will eat the chlorophytes that are there, and that creates a clear water phase, and that's usually followed by, either more green growth or additional cyanobacterial growth in the summer, or like early fall when temperatures are high and there's more nutrients available. So that's just kind of like your typical seasonal succession. That's not always the case in every system, though. It really depends on the phytoplankton community structure in that system. So in some places, like oligotrophic systems in particular, there's not always like a pronounced peak of biomass for all of your algal groups, or necessarily like they're not always super prominent relative to one another. Yeah, go ahead, Lawrence.
Yeah, hi. So I was just wondering, did you observe any changes or phenotypic plasticity when it comes to the cyanobacterial groups? Like for example, in co-evolution with the glaciers, did you observe any changes on the morphology of that particular species of the atoms? Oh, sorry, particular species of cyanobacteria in your study. So did you encounter any morphological changes when it comes to
I'm
co-evolution research?
I'm having a little bit of trouble hearing you, Lawrence.
Ah,
Sarah, are you able to hear better, or if you can put it in the
Yeah,
chat, Lawrence?
yeah, I'm having trouble too, Lawrence. Maybe you could write
sorry.
your
My,
question.
yeah, maybe my volume is a bit lower. Sorry for
It's
that.
like kind of coming in and out. I can hear you, but it's kind of like the volume is coming in and out.
Okay, no problem. I'll just take the questions.
Thanks.
Maybe while Lawrence is writing his question, um, Kate, I'm curious, having worked on chlorophyll and also sort of chlorophyll concentrations in lakes and rivers, and also the phaeophyton as the degradation product of chlorophyll. Do you consider that at all when you're looking at loss processes to monitor the development of phaeophyton as a marker of a bloom decline?
That's really interesting. No, I haven't, but I think that's a really kind of cool and unique way of thinking about that. I feel like most often when I think about phaeophyte, I'm just thinking about it like, was it corrected or not corrected for in chlorophyll, which is, I would imagine, like some of the stuff that came out of the work that you were doing, but not necessarily. I just don't think that like loss is really kind of like tracked at an ecosystem scale or like in situ, really. There's a lot of this I think is culture studies, not a lot of kind of like tracking in the field, like And I think it would be really logistically hard to do that, is like how much like quantifying these loss processes like in situ, or like, you know, in an actual ecosystem.
Mhm.
Just kind of like you might be able to note what processes are occurring, but not necessarily like more of a qualitative than a quantitative approach to that. But I wonder, yeah, maybe if the phaeophyton would be a way of looking at that. Um, Lawrence has his question here. Did you observe any changes in the morphology of cyanobacteria in grazing or viral infection? So we didn't actually study, this was a review paper, so we were just kind of like pulling information from others. So that's not something that I specifically observed, but I can say that I've worked on other projects looking at like species from, so I'll take like species of cyanobacteria from Lake Superior, and then I'll take them into the lab, and they look completely different from when I took them out in the field. And, you know, maybe I just like expose them to nutrients and temperature, and the morphology and the structure of them will completely change. And so they're almost like a different strain by the time you get them under laboratory conditions. So I imagine that plays a role in the grazing and viral infection, but I don't know if there's a change in morphology in response to um the grazing or infection in particular. But I would imagine maybe for the infection there is, if the cell doesn't die.
Thank you. Well, we got one more question. This will be the last one since we're at the top of the hour.
Yes,
Yep.
yes, yes.
Go ahead, Eldrin.
I just have last one question about the macrophytes, because they have like allelopathic activities to cyanobacteria. Can I say that a body of water with macrophytes is less likely to have algal blooms as compared to those without or with less macrophytes?
I think it's less likely, yes, because not necessarily because there's always allelopathic chemicals, but just because there's more competition in the water column for nutrients, and you might have like a more complicated food web. But I can't say if that's like a hard and fast rule that if you have macrophytes present that you're for sure less likely to have cyanobacterial blooms. But I think just like quickly thinking about it, that tracks to me because of the additional competition for nutrients in the ecosystem.
Yes. Okay, okay. Thank you.
Thanks, Eldrin.
Okay. Yeah, I'd like to thank you again, Kate, for a fantastic talk, and thank you everyone for joining. I put a link to the Harris et al. Paper in the chat, so go ahead and read this paper on loss processes. And Kate, if it's okay if I invite people to contact you further with further questions if they didn't get to ask their questions.
Yeah, of course. And if you're interested in anything regarding Glion, feel free to shoot me a message. My email is up on the screen here, or I can throw it in the chat as well.
And stay posted to the diatoms.org website. We will post the recording of this talk in a couple days. It might be a week or so, where we'll have a special page for the recording and information on the talk. And please join us in 2 weeks on September 24th. We'll have Juan Pirela Karlusic from MIT, who is going to speak on global ocean patterns of planktonic diatoms. So I hope to see folks again in the next talk in this series. And thanks again, Kate, and thank you to everyone for joining today.
Thanks, everybody.
Take care.
All right. Do you need anything else from me, Sarah, before I hop off here?
That's it. I'll follow up in an email because I have lots of things to chat with you about. So
Sure. Yeah,
yeah,
that would be great.
appreciate
Cool.
it. Thanks so
Yeah.
much. Take
Yeah,
care.
you have a good one. Bye.
Bye.